Today's lecture is a continuation of Chapter 18. So, epithelial cells line our intestines, and they only hang around for a few days and they die by apoptosis. So the new cells then are derived from the continuous but slow division of stem cells. So we're going to talk a little bit about stem cells in this chapter, and they're at the bottom of what we call intestinal crypts. So if we look at our intestines, you know that they have villi, microvilli, and down at the bottom here we have this part would be called the crypt. As you go down at the bottom, those purple cells there are the stem cells, and then we get shedding that occurs at the top of those. So if you start then from the bottom with the stem cell, it can start to amplify itself, and then actually with different signals, it can be your absorptive type of epithelial cells, goblet cells, or enteroendocrine types of cells in your intestines. So stem cells also play a role in repair of damaged cells, and if you look at our skeletal muscle, they don't have a whole lot of turnover, but they can regenerate rapidly in response to injury or exercise. And then we have what are known as satellite cells, and those are stem cells of adult muscle, and they're normally arrested then in G0, but they will proliferate in response to injury. So most adult tissues have stem cells which reside in distinct microenvironments or niches that such as the intestinal crypts that we already talked about. And they provide signals that mammalian stem cells throughout life and control the balance then between self-renewal and differentiation. Now, identification of the stem cells and their niches is a pretty big challenge these days in stem cell biology. The stem cells in intestinal crypts were first found in 2007, and we found these, they're called Wnt polypeptides that are secreted by adjacent cells for epithelial and fibroblast control. So what happens then is we go down to the crypt again, our stem cells are down here. So Wnt actually will signal then for these cells to start doing some other things. Now, again, there's potential for adult stem cells to be useful in clinical work. Basically, stem cells could be used to replace damaged tissue and treat a lot of disorder, diabetes, muscular dystrophy, Parkinson's, etc. The best type of stem cell would be embryonic stem cells. They have what we call pluripotency, and that means that they are able to develop into all of the different types of cells in human tissues. So there's a huge interest in embryonic stem cell research. Now, they were first cultured in 1981, and there's a lot of controversy when it came time to actually look at human ones, because people felt that we were harvesting embryos, fertilized eggs, just for research purposes alone. But the thing is, they can be propagated indefinitely. They can also be used to introduce altered genes, provide model systems to study cell differentiation. So this was a good tool then, or a precursor to what we actually see in humans today. So please take a screenshot of this slide here, guys. And if you remember a little bit in Bio 1, when you talk about development, so you've got a blastula and you've got a gastrula, etc. So within this mass here, guys, would be your stem cells from the embryo. So it wasn't until 1998 that first human cell lines were established. And again, this raised the possibility of using them then in transplants and other types of therapies. So in therapeutic cloning, the nucleus from an adult cell would be transferred to an enucleated egg. So that just means that the nuclei was taken out of the egg. And then the embryo then could be differentiated for transplant purposes. So here's your unfertilized egg. You take the nucleic acid out of that. Then you take the nuclei from an adult cell, stick it in culture, and get your embryonic stem cells that way. And again, then you give the right factors, basically, to make the cell types that you want and transplant that back into the patient. A lot of problems: low efficiency of getting the embryos. By somatic cell nuclear transfer, only 1 to 2% survive. Ethical concerns, again, cloning humans, a lot of people are afraid that's actually what's going to wind up happening if we continue with this. The difficulties can be overcome by what we call induced pluripotent stem cells. And basically that's reprogramming somatic cells to resemble embryonic stem cells. There's only 4 transcription factors introduced by retrovirus vectors that are needed to reprogram adult mouse somatic cells. So basically, here's your adult mouse fibroblast. You infect it with a virus, basically, that has the genes that you want. And then this induces this cell then to become what's called a pluripotent stem cell. Now, adult fibroblasts can also be reprogrammed to become pluripotent. And again, this is a new way then that we can try to do stem cell research and use that for transplantation therapies. So there's a lot of different combinations of transcription factors that people are studying. They activate different types of proteins and enzymes that are expressed in the embryo. And some of the factors that are used are called OCT4, SOX, and NAGAG that play central roles in this program. So they've isolated these factors and they're found to be pretty good. So they form an autoregulatory loop, and that positively regulates then the expression of the others. So they also activate other genes that maintain this pluripotent state. And at the same time, they're repressing the genes that enable differentiation. So our little triangle here, guys, they will activate the pluripotent genes, and they will repress the differentiation genes over here. So again, this autoregulation maintains the pluripotency, and it will allow the cells to undergo differentiation. Again, it overcomes some of the obstacles we've had. There's a lot of them that are out there. Sometimes the transcription factors that were used were also oncogenes, and sometimes the actual retroviral vectors were causing some types of mutations. So the term transdifferentiation, please know, is reprogramming somatic cells into other types of differentiated cells. So fibroblasts, for instance, to muscle cells or to neurons. This then would bypass the need for pluripotent stem cells. And mouse fibroblasts have been turned into heart muscle cells, guys, and nerve cells using a couple of different transcription factors. So again, if you take a look here, researchers have actually taken an adult mouse fibroblast, added these, turned it into heart muscle, added these, turned it into a nerve cell.